For doing one tube osmotic fragility test, we need a special rack with printed material on its backside, 3 test tubes, and a reagent called OTOFT reagent. And of course, we need the blood samples. The blood samples are collected in EDTA, and we have to centrifuge the sample at about 3000 RPM for 1 to 2 minutes so that the red cells are packed at the bottom of the tube. Then we take the OTOFT reagent, approximately 2 ml of the reagent in each of the tubes, and you can fill in as many tubes as you want to. Then we'll take the sample, 25 microliter, from the bottom of the tube and we have to be careful that we don't take any plasma with it. And we'll add this to the tube and shake it a little bit. And then the next tube and the second sample. Again, we have to be careful while taking the sample. We have to take the packed red cell from the bottom of the tube. Mix it again and keep it here. Then we wait for about 5 minutes and then we take the reading. One tube osmotic fragility test is a test which is usually done to detect microcytic red cells, and this is a very useful test for population-based screening of individuals who have microcytic red cells. Now this feature is a common finding in individuals having beta thalassemia trait, and here I am going to show you 3 different types of results of this test. The tube on your left hand side is a positive result in which the individual has microcytic red cells which are resistant to osmotic lysis at a low ionic strength, and therefore you're not able to see through the contents of this tube, and these individuals should be further investigated by doing hemoglobin electrophoresis. The tube on your right hand side is a clear negative result in which, since there were no microcytic red cells, therefore the normal or the normochromic red cells have been completely lysed and you can clearly see through the contents of this tube. And these individuals who show a negative result can be declared that there is a more than 97% probability that they do not have beta thalassemia trait and they do not need further investigation. Now the result in the middle is suspicious or borderline result. This usually results from the presence of microcytic cells which are borderline. For example, MCV around 73, 75 or something like that. Or there could be some other factors in the sample which can give a false positive result. In any case, whenever an individual shows a suspicious result, we should take it further for further investigation and do hemoglobin electrophoresis to confirm whether it has thalassemia trait or it's just a false positive result.